I'm sure we've all seen birds flying over our heads as the weather gets colder, but have we ever wondered, how do they do it without food or water?
Welcome to McMaster Geographic. In today's episode, we'll delve into the metabolism of migratory birds.
The metaphor of marathon running is inadequate to fully capture the magnitude of long-distance migratory flights of birds. In some respects, a journey to the moon seems more appropriate. Birds have no access to supplementary water or nutrition during a multi-day flight. These birds are seeking regions with adequate resources like food and nesting. While some species will travel short distances to different states within a country, others are long-distance migrants and move to a complete separate continent. These migratory birds come in all shapes and sizes with remarkable metabolic capabilities. People have always been interested in how these birds are able to maintain their energy for long, non-stop flights.
There have been many studies that try to explain how the body's energy stores support the exclusive demands of long flights. These studies have a wide range of objectives, which include examining the food composition before the flight, determining the fuel selection during the flight, and testing for changes in protein expression within the migratory birds.
One study from 2005 showed that migratory birds rely primarily on lipid stores to derive energy during their flight. They used bar-tailed godwits as a model to determine the energy fuels associated with flight. Typically, these birds travel 4,500 kilometers to their single stopover site where they can refuel before continuing their journey. For the study, they caught the arriving godwits immediately after landing at the stopover site and evaluated plasma concentrations of 6 key metabolites. They compared metabolite levels between the just landed godwits to the birds subjected to 5 hours of inactive rest and to the refueling birds that had already been at the stopover site. They found that plasma-free fatty acids, glycerol, and butyrate were significantly different among the migratory stages. Specifically, the arriving birds had higher levels of free fatty acids, glycerol, and butyrate in the blood than the refueling birds. This suggests that there is a high degree of lipolysis or beta oxidation associated with migratory travel. The free fatty acids are released from triglycerides stored in the muscles and adipose tissue. They also found that in the arriving birds, the plasma triglyceride levels were extremely low. However, the refueling birds had significantly elevated levels. Once again, this suggests that lipids are the primary fuel during flight, since the triglyceride stores are almost exhausted in the arriving godwits.
Another study examined the effects of fatty acid composition on peak metabolic rates in migratory birds and whether fatty acid composition enhanced exercise performance. To do so, they captured red-eyed vireos and separated them into 2 different diet groups. Both these groups were fed a high lipid fruit diet, but the distinction between the 2 groups lied in the percent of unsaturated fat. One of the diets consisted of 58% unsaturated fat, while the other consisted of 82%. Mass-specific resting and peak metabolic rates were also compared. The results showed a predominant presence of monounsaturated phospholipids in sandpipers, along with a decrease in the N6 fatty acid levels between pre-migration and migration periods. What this suggested was that N6 was utilized in migratory flight. As for the metabolic rates, it was found that mass-specific resting metabolic rates for both the diet groups were relatively similar. What did differ, however, was the mass-specific peak metabolic rate, where the group that was fed 58% unsaturated fats displayed values that were 25% higher than the 82% unsaturated fat group.
Just like mammals, birds can synthesize saturated and monosaturated fatty acids, but do not have the enzymatic capacity to insert double bonds at the N3 or N6 positions on the fatty acids.
So this makes the pathway for the natural doping hypothesis. The natural doping hypothesis proposes that high intake of N3 polyunsaturated fatty acids increases migratory performance by enhancing the functional capacity of membranes and increasing the aerobic capacity of flight muscles.
The N3 polyunsaturated fatty acid content of triglycerides and phospholipids was positively correlated with the activities of citrate synthase and 3-hydroxyacyl-CoA dehydrogenase in flight muscle.
There lies a relationship between phospholipid composition of muscle cells and the oxidative metabolism associated with it. The fatty acid composition of membrane phospholipids can be altered by the diet. Results reveal that the consumption of N3 fatty acids modifies all membranes equally. Therefore, the changes in membrane fluidity, permeability, and N3-N6 ratio that result from N3 fatty acid incorporation are known to influence the activities of key oxidative enzymes and ATPases. These include citrate synthase, carnitine palmitoyl transferase, the sodium-potassium ATPase, and calcium ATPase, among others. Only extreme regimes of endurance training can lead to increments in oxidative capacity matching those induced here by diet in domestic quails. In preparation for long migrations, some birds improve their physical fitness by eating. When maximal energy storage is critical, choosing N3 fatty acid doping over endurance training strikes us as the better strategy to increase aerobic capacity.
They captured white-throated sparrows in both migratory and non-migratory seasons and removed their pectoral muscles for analysis. The expression of the fatty acid translocase mRNA was 100% higher in spring migrants and 170% higher in fall migrants than in the wintering birds. Moreover, the mRNA expression of the plasma membrane fatty acid binding protein was 80% higher in spring migrants and 70% higher in fall migrants than in the wintering birds. Furthermore, the mRNA expression of the heart-type fatty acid binding protein was 1,000% greater in migratory seasons than in the winter. All of these proteins are associated with fat metabolism. Specifically, they enhance fatty acid uptake across muscle membranes. Therefore, the fatty acids from the adipose stores are delivered to the muscle mitochondria for oxidation.
We also examined the activities of different enzymes involved in fatty acid oxidation. There was a 90% increase in the activity of citrate synthase in migrants compared to the wintering birds. Elevated citrate synthase activity, which catalyzes the first reaction of the TCA cycle, indicates that an increased level of oxidative phosphorylation will occur to power the flight. Similarly, there was a 90% increase in carnitine palmitoyl transferase 2 activity. This carnitine transfer system is important for transferring acyl groups across the inner mitochondrial membrane. Once inside the matrix, beta oxidation can occur to generate acetyl coenzyme A, which will ultimately be used to generate ATP. Compared to the wintering birds, the fall migrants showed a 30% decrease in lactate dehydrogenase, or LDH, activity, and the spring migrants showed an additional 19% decrease in LDH activity. Lactate dehydrogenase is an enzyme used for carbohydrate oxidation, especially during anaerobic conditions. Reduced activity during migration suggests that carbohydrates are not the primary fuel source for these birds.
Research on these long-distance migratory birds can have important implications for wildlife conservation. There are over 30 different migratory bird species that are critically endangered, and understanding how these birds function can give further insight on how to assist these species from becoming extinct. Preserving every species is essential for sustaining ecosystems, as the loss of a species may result in a cascading effect which can lead to further implications. This could result in negative effects on biodiversity, in which many people's livelihoods depend on.
Studying migratory birds can also improve our understanding of metabolism and exercise. This knowledge can help us to tackle problems in our society. One such example is improving the performance of athletes. Similar to these birds, athletes can benefit from pre-exercise loading and preparation. The timing and composition of meals can optimize metabolism and subsequently enhance performance. For example, the nutrient intake of carbohydrates leading up to competition has been proven to increase endurance. Also, the reliance on high-fat meals can cause the body to increase fat oxidation and spare glycogen stores. Further research of long-distance migratory birds, specifically on how they efficiently metabolize fat, can provide new information on how humans can increase their endurance performance by mimicking these magnificent birds.
To summarize, the question addressed today was how birds fuel up their mighty expedition during the winter. We covered the role fats play as a primary fuel source, the importance of unsaturated fatty acids, the change in the activity of different enzymes, and the implications and significance of the research.
The types of food consumed by these birds and the metabolic processes that take place can be applied to a broader field involving sports medicine and early-genic age. There hasn't been a lot of research in this field in the past few years, but a rekindling can help us further apply this to our society.
Tune in next week for another episode of McMaster Geographic at 5 p.m. to learn more about thermal regulation in polar bears.